gram-negative prokaryotic cell. The most distinctive feature of the Caulobacter Crescentus cell is its dimorphic life cycle. A dimorphic cell means that during cell division, the mother cell will produce two different types of daughter cells. One of the daughter cells will become a stalk cell, and the other daughter cell will become a swarmer cell. Section 2. Life of a swarmer cell. A swarmer cell has a polar flagellum, which means that it only possesses a single flagellum at one end of the cell. The swarmer cell possesses pili, which surround the flagellum. Specific membrane proteins called MCP proteins are also found embedded in the cell membrane surrounding the flagellum. The flagellum provides motility, while the pili help bring in nutrients into the cell. The chromosomes then become condensed inside the mother cell, which results in the formation of a stalk. Section 3. Life of a stalk cell. After the swarmer cell swims for about 30 minutes, the cell will begin its transformation into a stalk cell. This transformation begins when the nucleoid of a swarmer cell uncools out of its condensed form. From there, DNA synthesis begins. The flagella is shed, the pili retract into the cell, and the MCP proteins degrade, and the cytoplasm extends where the flagella was to form the stalk. The function of the stalk isn't exactly known. However, it is believed that it increases the cell surface to volume ratio to absorb nutrients, as well as to provide a source of attachment to other cells. The stalk tip contains an adhesive patch called a holdfast. This holdfast allows the stalk cell to attach to solid substratum, which is often another cell. The stalk cell is considered the mother cell, and as DNA is synthesized, the process of cell division begins. Section 4. Steps of cell division. Step 1. The chromosome is replicated, and the cell is almost twice its original size due to a septum beginning to form. Step 2. A flagella and multiple pili form opposite of the stalk, and MCP proteins form in the cell membrane above the flagellum and pili. At this point, there is a stalk cell and a swarmer cell attached to one another. Step 3. There are approximately 50 genes that are responsible for the transformation of a stalk cell into a swarmer cell. A special protein called CTRA activates transcription of these genes, which code for the polar flagellum to be made. Step 4. While the cell is dividing into the daughter cells, the nucleus of the swarmer cell is condensed, unlike the nucleus of the stalk cell, which stays extended. Chromosome replication then takes place in the stalk cell immediately after cell division. This allows the stalk cell to be prepared for another cell division. Step 5. FTSZ protein polymerize and form a ring around the cell. This ring provides and constricts and allows for cytokinesis. FTSZ protein is only present in the stalk cell after cytokinesis. Section 5. The conclusion. How is a dimorphic life cycle beneficial to a cell? Caulobacter normally live in very dilute places. Being able to attach to other cells can help their survival in these environments by allowing cells to exchange nutrients with one another.